Good day, students. Welcome back to biology class. My name is Fawe Babajide. In this class, we are still looking at microorganisms. Microorganisms. Now, so far here, we have discussed number one, the meaning of microorganisms. That they are tiny organisms that cannot be seen with our naked eyes except with the aid of microscope. Tiny organisms that cannot be seen with our naked eyes except with the aid of microscope. Then number two, we have discussed classification of microorganisms. Classification of microorganisms. That we can classify these organisms into different groups. Number one, we have what is called the viruses. Number two, we have bacteria. Number three, we have the archaea. Number four, we have the protozoa, the protists. Number five, we have algae. And number six, we have fungi. And as well, some normally include hermits in their own classification. Now, that's number two. Number three, if you remember, we also discussed some things about these organisms. Few parameters about them. For example, most microorganisms are unicellular, but some are multicellular, as you know them. So, we also discussed characteristics of microorganisms. For example, I said most are unicellular. Most are unicellular organisms. Yes, though some are multicellular. For example, viruses, though they are non-cellular, no cell. Bacteria and archaea plus protozoa, they are all unicellular. While algae and fungi are multicellular except yeast. They also said they reproduce asexually. People don't know what? Asexually. Either by binary fission or spore formation. Yes, that's another characteristic of all microorganisms. I also said some are parasitic, some are saprophytic, while some are what? Free living organisms, etc. Remember, I also gave us that some are pathogens, some are beneficial to us. Now, we have also discussed, still under this, concept of culturing. Concept of culturing microorganisms. That culturing simply means techniques involved in growing microorganisms in our laboratory under certain conditions. We can grow these organisms in our laboratory under certain conditions. We provide the environment that will enable them to grow, the mineral requirement they need. So we do that using what is called culture media. Culture media. Media, plural is medium. A medium is a combination of nutrients used in growing microorganisms, which can be liquid or gel. Now, and I said from here that we can have culture for growing cells. So we call those ones cell culture. And we can also have the ones for growing microbes. What do we call those ones? Microbial culture. Microbial culture. Yes, under this again, I also gave us steps involved in preparing culture. That's preparation of culture. You still remember? Preparation of culture. That the steps involved weighing the medium is actually, for example, potato dextrose agar. You weigh it, then you dissolve in water. What's the next thing? You sterilize. This is what is called an autoclave. Then you allow it to cool down. Then pour in your petri dish. And the next thing is what? Inoculation. Inoculation means the introduction of the desired organisms on the plates. Then after inoculation, we now do what? We incubate so that the organisms can grow at the optimal temperature. So then after you have done that, what's the next thing we now say? We now try to identify that identification of organisms. Yes, using some parameters like you can use microscopes to identify them. We can use biochemical properties. We can use the forms of the colonies, or we can use the staining techniques like gram staining techniques. As well, we have discussed ways by which organisms enter our body. We have different ways. Simply say they are undivided, they are everywhere. We have the skin, we have the mouth, we have animal bite, we have direct contact. And as well, in another class, we also treated carriers of microorganisms. Carriers of microorganisms. Where we said we can have living carriers, which we normally call vectors. And as well, we can have non-living carriers, though we call them those ones are also called carriers. Non-living are called carriers. Why this one involves living things. Living things. This one involves non-living things. Okay. Now, I also give you examples of this. Today, we want to briefly look at microorganisms in our environment. Microorganisms in our environment. And what does it entail? What does it entail? Number one is that these microorganisms, as we all know that they are tiny organisms, they are everywhere. So there is a term we use for that microbes are unbelievers. Meaning they are everywhere. Yes, you can see them in what? In air. Microorganisms are what? They are abundant in the air. They don't grow in the air, but the spores are dispersed in the air. So when the spores now fall on the suitable substrate, what do? They germinate, grow, and produce another organism. Example of organism in the air, you have something like influenza. You have all these viral particles in the air causing something like measles. And now we know this one's common, this COVID-19. They are all examples of airborne diseases. Now, microorganisms are also found in water. Yes, because water is rich in both organic and inorganic nutrients. So we can see them in water. Examples, we have this Clostridium species. We have this Vibrio, etc. They are all found in water or contaminated food. Then we can also see them in the soil. They are found in the soil. For example, Clostridium tetani or Clostridium winchi. Clostridium tetani, that's one causing tetanus, or Clostridium winchi. They are found in the soil. They are abundant in the soil. You know, they aid in the recycling of materials in the soil. They aid in the decomposition of the organic matter, returning the nutrient back to the soil. While some are also harmful, so they are found in the soil. They are also found in our food substances. That when, for example, our food is rich in pH, moisture content, and nutrient, microorganisms are also found there. Now, so these organisms can be isolated from different environments and they can be grown in our laboratory. Exactly. So we can get them from the environment and then grow them in our laboratory. So that means we can easily look at what's called growth of microorganisms. Growth of microorganisms. Like I told you that microorganisms are grown in the laboratory using what? Our culture media. Culture media means combination of nutrients. So this culture media is are used to grow the organisms. So we have different types of this culture media. Different types. It depends on the interest of the microbiologist. For example, we have types of culture media. Culture media. We have number one, what is called selective media. These type of media are used to grow organisms of interest. When it's cultural media, it does not permit other organisms to grow. This one selects certain organisms to grow in them. Examples are monitor salt agar. Yes, monitor salt agar. Is that clear? Some organisms that can contaminate will not be able to grow. So it selects organisms to grow. We also have another one called differential media. Differential media are the ones that usually they are used to distinguish a particular colony. Example, when you have a culture can be mixed or pure. Pure means it contains only one colony of interest. While mixed means you have different populations. So but this selective means it will give you something like pure culture. But in case of differential, many organisms can grow on a bed. The one of interest can easily be identified, probably through the color, or forms or structure. We also have an example of this one, microbes agar. There's something called microbes agar is an example. Microbes agar, although it's also a selective medium, there's also used as a differentiator. Now, here, you can also have what's called enriched media. Enriched media. Now, what's the meaning of this enriched media? The enriched media usually enables organisms of interest to grow faster than others. This one contains nutrients that facilitate the growth of the desired ones to grow faster than the other ones. Examples are driven by the agar, which is used to grow certain organisms. So we have different types of cultural media that can be used to grow organisms in our various laboratory. Now, the next thing here is now what is called growth requirements. For organisms to grow, growth requirements. To grow, they need certain materials, which can be chemical requirements or physical requirements. Chemical requirements or physical requirements. Chemical now, in terms of mineral requirements by the plants, by the organisms. They require something like carbon. Most of these macromolecules contain carbon. For example, our carbohydrates, our lipids, our proteins, they all contain carbon. The carbon is the backbone of organic compounds. So they need carbon, they need nitrogen for protein synthesis, amino acid synthesis. They need phosphorus and other things. Some of them need oxygen, the aerobes, while anaerobes need oxygen. So all these are chemical requirements by plant. If those things are not there, then it is difficult for some organisms to grow. The physical requirements, we'll be talking about something like the pH, the temperature, the osmotic pressure, and etc. If those things are provided, then it will aid the growth of these microorganisms. Now today, we have been able to look at microorganisms in the environment. Microorganisms are everywhere in the environment. You can see them in the air, you can see them in the water, they are found in the soil, they are found in our food, etc. They have mentioned something like growth of microorganisms. They can be grown in our laboratory using what we call cultural media. As how we have different types of cultural media: selective, differential, and enriched media. And at the same time, growth requirements. They need chemical requirements and physical requirements. Join me in the next class where we'll be discussing growth curve of these organisms. Thank you for watching. Stay blessed.